The overall goal of this experiment is to longitudinally evaluate the macrophage infiltration within the synovium in a rabbit model of septic arthritis using MR imaging. This could be performed with the use of iron oxide particles that label macrophages and reveal their presence. Macrophages are key cells in the development, amplification, and resolution of arthritis, especially in septic arthritis. The in vivo demonstration of their presence and distribution within an infected joint could be a valuable tool to monitor antibiotic therapy, as an healed joint is not supposed to be massively infiltrated by macrophages. Macrophages can be labeled using nanoside MR contrast agents such as USPIOs, ultrasmall superparamagnetic iron oxide. After intravascular administration, they are specifically taken up by activated macrophages. USPIO are composed of an iron oxide core with a crystal size of about 14 nanometers. They act as small magnets that generate MR signal changes within macrophages containing tissues. You can see on these two axial MR images of a rabbit knee that 24 hours after USPIO administration, the synovium presents an intense and diffuse signal loss due to the presence of USPIO-loaded macrophages. The number of dark pixels observed on MR imaging is correlated to the iron content within the tissue and therefore to the number of macrophages. The longitudinal follow-up of USPIO-related MR signal changes could therefore give an in vivo evaluation